Have you ever played a puzzle game? It's the game in which pattern arrangement and pattern recognition is involved. But if any single block is not arranged in correct position, it will cause disarrangement of the whole game. Keeping this concept of placing puzzles at right place, let's understand. What are the types of genetic alternations? Well, mutations are permanent modifications in the base sequence of DNA. They can occur at the level of one or a few bases of DNA as point mutations involving substitution, deletion, or insertion, substitution of a purine by another purine is called as transition, or of a pyrimidine by another pyrimidine is also a transition. In other case, exchanges of purines and pyrimidines are transversions. At the level of a gene, mutations involve dozens to thousands of bases. At the genomic level, mutations include deletions or duplications of hundreds of thousands to millions of bases up to chromosome rearrangements and aneuploidies. Moving forward, copy number variation involves large deletions and insertions of various lengths created by unequal crossing over between misaligned segments of repetitious DNA, or by non-homologous end joining. Unequal crossing over is the origin of X-linked anomalous color vision. Activation of enhancers and silencers can cause phenotypic variation in expression of the genes they control. Dynamic mutations involve expansion of triplet repeat sequences and can undergo further expansion or contraction from generation to generation. Now let's discuss these genetic alternation, which are substitutions, deletions, insertions, frameshifts, and duplications in detail. Let's discuss silent mutation, which is a type of substitution. Substitution involves replacement of a base pair if the amino acid encoded by the new codon is the same. It is a silent mutation, as this is the normal sequence, having stand, anti-stand, and they encode these amino acids. Now thymine and adenine are substituted, and cytosine and guanine are removed. It is called silent, because both thymine-cytosine and thymine encodes for phenylalanine. But in missense mutation, substitution involves replacement of a base pair if the amino acid encoded by the new codon is different. Let's understand it. This is normal sequence. But in missense mutation, cytosine and guanine replace thymine and adenine, but in this case different amino acid is encoded, which is lysine. Some missense mutations do not alter the chemical properties of the protein, is called as conservative mutations. For example, change the nature of protein from hydrophobic to hydrophilic, or change the nature of protein from hydrophilic to hydrophobic. Hydrophobic means water-hating property, but hydrophilic means water-loving nature. Whereas others have a deleterious effect. In some cases, though, heterozygosity of a deleterious mutation may create selective advantage. A notable example is the substitution of the 6th codon in the beta-globin chain responsible for sickle cell anemia. which in heterozygotes confers resistance to malaria. Let's understand it. A normal hemoglobin has DNA sense strand, messenger RNA that encodes normal beta-globin. A 6 codon encodes for glutamic acid. But in sickle cell anemia, there is substitution of thymine and uracil. Now it encodes for valine instead of glutamic acid. Sickle cell anemia is an inherited disease in which the red blood cells have an abnormal crescent shape, block small blood vessels, and do not last as long as normal red blood cells. Other type of alternation is premature termination or nonsense mutation. Well, substitution can create a stop codon, causing translation to come to a premature halt. This is called a premature termination or nonsense mutation. This is normal sequence. Codon at position 4 encodes arginine. But in case of premature termination or nonsense mutation, thymine and adenine are substituted in place of cytosine and guanine. Now it encodes for stop codon. Next is single base pair deletion, causing frameshift, in which from normal sequence, single base pair deleted are removed. This causes frameshift. Let's understand it. This is normal sequence. Second codon encodes for phenylalanine. When thymine and adenine are removed from normal sequence, it causes frameshift. Due to frameshift, different amino acids are produced. Moving forward to single base pair insertion, also causing frameshift to the normal sequence. Guanine and cytosine are added, leading to frameshift. So different amino acids will be produced. So duplications of whole genes can lead to disease. For example, Charcot-Marie Tooth disease. This is a peripheral nervous system disease involving progressive degeneration of peripheral nerves, leading to atrophy of distal limb muscles. It can be caused by a variety of types of mutation, but about 70% have a large duplication of chromosome 17. That includes the gene PMP22, encoding a peripheral myelin protein. This contributes to demyelination, whereas deletion of the same region causes a paralytic response to pressure. If a deleted or inserted segment is of other than a multiple of 3 bases, the translational reading frame is disrupted in a frameshift mutation. The most common mutation causing Tay-Sachs disease. It is a 4-base insertion causing a frameshift and leading to premature termination, so that no functional hexosaminidase A is synthesized. Let's understand it. This is normal sequence. That causes functional hexosaminidase A to be synthesized. But in Tay-Sachs disease, 4-base insertion, frameshift, and premature termination occurs. Tay-Sachs disease is a rare genetic disorder, passed from parents to child. It's caused by the absence of an enzyme that helps break down fatty substances. These fatty substances, called gangliosides, build up to toxic levels in the brain and spinal cord and affect the function of the nerve cells. Moving forward, cystic fibrosis is an example of 3-base deletion from the normal sequence. 3 bases, which are cytosine, thymine, and thymine, are removed, or their deletion occurs, leads to removal of phenylalanine. Moving forward, 50% of severe cases of hemophilia A affects the activity of clotting factor VIII, involve a novel mutation called a flip inversion. This occurs as a consequence of 2 unusual conditions. First is the existence of several copies of a small gene called A located near the XQ telomere, plus another within intron 22 of the F8 gene, and the fact that the long arm of the X chromosome has no pairing homologue in male meiosis. As a consequence, internal recombination sometimes occurs, following looping back of the end of the X, breakage, and rejoining within the A genes causes inversion of the intervening segment of the F8 gene, leads to flip inversion, and production of hemophilia A gene. Hemophilia is usually an inherited bleeding disorder in which the blood does not clot properly. This can lead to spontaneous bleeding as well as bleeding following injuries or surgery. Blood contains many proteins called clotting factors that can help to stop bleeding. Moving forward towards the nomenclature of mutations. Mutations are named by conventional abbreviations and may be described by the change at the DNA or protein level. For example, the most common mutation responsible for cystic fibrosis is a deletion of 3 bases that encode phenylalanine. In the coding sequence of the gene for the cystic fibrosis transmembrane regulator, and the codon position is 508. So this was all. If you need to learn more about this topic, do watch our video on genetic code. Well, hope you have a better understanding of the types of genetic mutations. Thank you for learning. Join us again.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.